talk about is how we used a large animal model here, the non-human primate, and rodents to look at specific disease. And this disease is asthma and rhinitis in the context of how they're affected by air pollution. So we've been fortunate, I've been fortunate over the years to work with many extremely talented collaborators, and we've had funding for over 30 years from various funding agencies, from EPA to NIH, and have used, also just recently we have had a large center grant that was looking at the health effects of air pollution using both laboratory animals but also human subjects. And I want to focus today on this problem of what is the best models, animal model. First of all, that maybe is not a very good term. So the term model, maybe we should be thinking about experimental model and disease models like Charlie talked about, and then look at the appropriate model to use rather than to starting with a mouse or a primate or a farm animal. And so the reason for that, as a pathologist and anatomist, I want to really target in what is the best anatomy that mimics the subjects that I want to make progress in understanding either disease or prevention or treatment. And so these challenge questions that we were given were really good ones, I think. And we've already heard that Dr. Eiland really did an excellent presentation about the advantages of large animal models over the rodents. I'm going to bring that in too, but also how we can use rodents to also to provide us information, and then we can go back and forth, but really concentrating on the disease. And then what are some of the changes working in these or challenges working with these models that we have to overcome? And there, I'm sure a lot of you already know that. I'm going to kind of talk about all of these throughout my talk. Also show how important infrastructure is here. And I couldn't have done any of this work if I didn't have the proper infrastructure to conduct it, especially with non-human primates. And then also the final thing is, how do you see these models increasing in coming years? And I'll try to bring this back. There's kind of a history of my journey, starting out as a veterinary student, all the way through where I am now. So I'm not going to take 30 years to give this, but I'm going to cut this down really quickly. But what I'm going to talk about, first of all, identify the problem. Look at comparative airway responses to air pollutants. I'm going to use just one, ozone. And then I'm going to show you about some new paradigm that we have learned over the years, especially very recently, about certain cells that may control it. And then the future for using either non-human primates or maybe if there's other animal models that we'll talk about. So this diagram kind of shows you how we started, how I started. Actually, I started with a large animal model, the non-human primate, and identified effects caused by air pollution on the airways of these animals, then carried the research into laboratory animals, and eventually, and I'll show you how I think we need to move it back into a large animal. But ultimately, it's not about us in here. It's about, as was emphasized in the previous talk too, it's about the condition or the person that we're trying to help through our research. And I think that always has to be the focus. And I'll show you this translation, as Dr. Eiland pointed out, this is the major gap. We haven't done a very good job in that. We create lots of data, but we don't translate it to the human condition or to laboratory animal disease. And there's a tremendous gap there that is been identified. So what's the problem that I've been studying? And that's the problem of air pollution. And this continues to be a major problem. Close to almost a half of people in the United States still live in counties where there's unhealthy levels of ozone and particulate matter. This happens to be in LA. But the health burden is tremendous. And this just illustrates percentage of mortality that that happens in different areas of the country. And you can see in the Midwest, in the Southeast, and the East Coast, and the West Coast, that's where the primary health problems are. And if we look at similarly where we're out of compliance for just one pollutant, like ozone, this kind of matches up. And so you can see that there's a huge problem of air pollution still in Los Angeles area and out in the east, and even here in Michigan and certain areas, counties. But the even more dramatic problem is in China and India, and where this has been like 4,000 people die per year, per day, something related to pollution. So this problem is actually been identified in India now, and actually it's about 3 times the problem that we see in China. So I'm going to take just an example now of how we use laboratory animals to understand the disease process and show you how it's related to one of the more tragic diseases of asthma. So we're going to talk about ozone and its health effects. This is an oxidant gas. It's found in photochemical smog. It's created as a secondary pollutant. And as I pointed out, many people live in areas where the concentrations are higher than they should. And this causes respiratory pulmonary function deficits, air inflammation, and airway inflammation remodeling. But it also causes cardiovascular diseases. Now it's been identified, along with particulate matter, reproductive diseases, neurological diseases, as well. And so it's sort of the new cigarette smoke where you have all these systemic problems due to this pollutant and air pollution in general. I'm going to talk a lot about asthma in the next few minutes and rhinitis. And I know you can't see it, everyone can see this, but this is kind of a mechanistic framework of how this air pollution affects asthma. Let's start down here. Here's asthma, and we know for years that air pollutants can exacerbate the condition. And so that's not really that's certainly we have to reduce the air pollution, this exacerbation. But we also understand now that pollution can also affect the onset of asthma, or even the development of asthma. And I'm going to talk about what we have learned from these animal models about this aspect. And actually, John Baum's a friend of mine who wrote this in Lancet. He emphasizes pollution in certain areas, but I'm going to try to show you that there's a totally different phenotype in terms of inflammation immunology that we have learned, that we have to understand better in children, but I think we can do this through large animal models as well. So the focus is then going to be, this talk's going to be on asthma. It's the most common chronic respiratory disease in children. 6 million children have asthma in the United States, 17 million asthmatic adults, and 300 million worldwide. But it's not just one disease. So it is actually now described as many phenotypes. And the emphasis of this research now is really to target what type of phenotype the individual person has. So asthma is looked at as a reversible airway obstruction and symptoms of wheezing. The pathology shows excess mucus and airway remodeling, that there's actually increase in the number of cells, and also fibrosis around the airways. But it's a reversible condition, but chronically. So it comes and goes. But there are some phenotypes that have certain cytokines, like what we call type 2 cytokines, that are very high in their expression. That leads to a number of pathologies in the airways I'll talk about. But there's other phenotypes that are very low, type 2. And it's not just allergic asthma, but also non-allergic. In fact, we heard a seminar speaker here last week from the UK that said that in children, at least 40% or more are non-allergic asthma that he sees in his practice. So there's early onset, late onset, there's different inflammatory cells, and these are now all identified as different phenotypes that we have to characterize. And the emphasis in this paper and several others is that we have to have better animal models to look at this, because we can't do it in the human subject. So this is actually some material that I got from the National Primate Research Center site, and it also emphasizes this emphasis in the United States and elsewhere on laboratory animals, and very few studies in non-human primates or other species like farm animals. The monkey is a good model. Mainly, it's been emphasized that because of its genetics, that we share over 90% of our genes with the non-human primate. And there are certain areas, and it's not one that was organ transplant, as well as looking at heart disease, brain disease, cancer, lung disease that I'm going to talk about, have been the focus of this large animal effort by these national primate centers. And in the United States, we have 7 of these centers, and one of these centers is at the University of California at Davis, where I trained, but also over the years I've had a number of opportunities for funding, because it is the only center, primate center, that has facilities for respiratory research. And that, what I mean by that is it has certain equipment designed for exposure of these large the primate. So let's just look at the primate and just talk about why and why not it might be a good animal model for asthma. One is the airway structure and function is very similar to the human. The anatomy, the cell biology, and dosimetry. So I just caution all of you when you're looking at your model, and if you're modeling for an animal model, if you're using that as a model for human, understand the anatomy of it, the molecular biology behind it, and really concentrate, first of all, on understanding the animal that you have for the particular disease. And that's what we have tried to do for this, and also in the rodent. The models of asthma are similar, more similar to the human disease than in the rodent. For example, the rodent model is often very short. It's a short exposure to an allergen, and you get some information on the response. But the large animal allows you to look at chronic disease, where you have much more of the phenotypes that you see in the human. It's also, as been pointed out before, in this there are better models for lifespan studies, and there's a tremendous emphasis in respiratory research to understand the effects early on in the unborn, all the way to the aged. And this is why this large animal model is a good one, I think. There's also more heterogeneity. In the age of, we're going to see a lot of effort being put into precision medicine. This is going to be the future. It's going to close lots of gaps, I think, in translation. And we need to have models where there's lots of variation that mimics the human condition. So let's just look at the anatomy between the mouse and the monkey. And I just point this out, these diagrams are just of the respiratory tract, the nose, the trachea, and then into the lungs. And this is a cartoon of the rodent. First of all, you can see that not just size, but also the anatomy can be quite different. There's only mainly down to the airways, what we're talking about, one major difference is the human has many generations of conducting airways called respiratory bronchioles. And this is the target for, one of the targets for ozone. And this, in the rodent, there are no respiratory bronchioles. So just anatomically, this model is very different. But if you look at the, this is the, look at the, just the size of the epithelium that lines these airways in the large upper conducting airways, the monkey epithelium is very thick, and it has lots of mucus cells. And in the mouse, there's hardly any mucus. Goblet cells is a different cell type called the serous cell. It's very thin, watery type of mucus. If you get down to into the lung, where the conducting airways meet the alveolar parenchyma, where you have a gas exchange, there too there's a major difference. So there's a much thicker wall of in the conducting airways in the deep lung compared to the rodent. And there's also large bundles of smooth muscle and a much smaller amount of smooth muscle in these terminal bronchioles in the rodent. So even why that's important, especially in respiratory research in air pollution, is that the pollutant actually targets the respiratory bronchioles and deep into the lung. And so here's some studies that we did a long time ago looking at the effects of ozone on these respiratory bronchioles. So we opened the lobe, dissected the airways into the exact same generation of airways in the alveolar or the respiratory bronchioles, and then characterized the response, and did that with all the animal models that are animals that we did, and characterized the epithelial and inflammatory responses. And after long-term exposure to ozone, you can see this thickening and corrugation of the airways that lead to pulmonary dysfunction. Well, the same changes, I'm not going to dwell on this, but also in the upper respiratory tract. And so in the human, the monkey is more similar to the anatomy in the human. It has, first of all, has a large amount of respiratory epithelium, like the human, and very little in the way of olfactory. In the rodent, half of the nose is covered by olfactory epithelium. So again, I'm just emphasizing the anatomy differences in these two species and the similarities. And we then went on to characterize all the epithelial dimensions of where the epithelium lies in the nose, all the way down to the conducting airways in the lung of the primate. And if we look at the response to ozone, we see this loss of respiratory cilia that lines. This is a carpet of cilia looking at scanning electron micrographs. This is a normal. After ozone, you lose the cilia. This is in the upper airways in the nose of the monkey, and then there's an increase in mucus goblet cells. And this feature here, I will come back to this later, but this increase in mucus goblet cells and thickening in an inflammatory response is the key also to asthma and rhinitis that I'll talk about a little bit later. But then we use other technologies to actually image the whole and reconstruct the airways. So this is just an example using the nasal cavity in the non-human primate. These were infant studies. We did computer or MRI imaging. We then reconstructed it, computerized 3D reconstruction, made these computational meshes, took the pathology, and then used morphometric techniques to not only identify the regions in the nose, but also the amount of cells of the different types. So we could overlay this now on this model. And then we can simulate the exposures in these animals and look at predicted areas where the ozone would hit the airway. first studies in the monkey, and then started looking in the rat, and then people picked up on this, and this was the first article that showed the same nasal lesions in the human. This is a study that was done in Mexico City by a pathologist, William Calderon. And interestingly, in all the species, which is a rarity, we found all the same effects. Then we went on to characterize this in the rat over a long period of time at different concentrations. And this just shows the increase in the thickness of the epithelium that line the conducting the nasal airway, and the increase in mucus. So what happens in both the primate and in the rodent, that you have this initial injury by the pollutant, ozone. That causes an inflammatory response with increase in neutrophils, and that led to this metaplastic response. This response is a characteristic of asthma pathology, where you get lots of increase in mucus. But then we started to look also in mice, and I just wanted to show you some epidemiology associations that in just the last few years have been shown that there's an association with high ambient concentrations of ozone in the onset of asthma in a large study that was just conducted in non-atopic or non-allergic Latino children. And also, there was they showed that ozone causes eosinophilic inflammation, another inflammatory cell type, the eosinophil, that's often associated with asthma. This is also increased in children, but interesting, not in children with allergy, but children without allergy. And then finally, we started to do using mice after repeated exposures to ozone. We identified eosinophil rhinitis and nasoepithelial remodeling, and then I better understood type 2 inflammation. So let me, in the remaining minutes, just show you what we have found and how we need to carry this back to the large animal model. So two of my students, one of the resident, Dr. Bing, is now back in Singapore, and Dr. Kumagai, who was a visiting scientist from Japan, really did the work to identify the pathogenesis of ozone-induced airway injury, and identified a really important fact, and that is they identified the cell type that we believe is actually the driver of almost all of these changes, except for the early epithelial cell death. So all this remodeling that takes place after 9 days of exposure that we saw in these animals, this increase in eosinophils and mucus, were all driven by innate lymphoid cells, and a specific type called LIMP2 innate lymphoid cells. So what are they? These cells are a newly discovered family of immune cells that do not express the lineage markers of T cells or B cells or dendritic cells or macrophages. These cells mirror the phenotypes of T cells. So in asthma, we're often hear of type 2 helper cells, but I'm going to show you that response isn't driven by these T cells with asthma in this asthma phenotype, but it's driven by these innate lymphoid cells. And unlike T cells, they do not express the antigen responses or undergo clonal selection that you would see with an adaptive immune response. But they respond quickly and actually in release cytokines that are very, very similar in more of these cytokines than what you see in the T cells. So the ILCs respond to the injured tissue. There are alarmins that are specific ones, or what we call epithelial cytokines, that are released and stimulate the ILCs, and they produce the cytokines that change this epithelium. So these ILCs play a role in pathology, and there's a number of them, different types, and they're actually types that also mimic the T helper cells. But we're going to concentrate on these ILC2s because they've been associated in humans and in mice to allergy and to asthma. So what happens is that we believe is the ozone damages or signals this airway epithelium, releases these larmins or these epithelial cytokines. We believe interleukin 33, IL-25, or TSLP are really the main drivers here. They stimulate the innate lymphoid cells. They can also stimulate dendritic cells, but this pathway of adaptive immunity, we just don't see this with the ozone response. And so what happens is these ILCs can then release the cytokines that increase the mucus that I showed you earlier that was shown in the large animal model and in the rodents. And this causes non-atopic asthma. Let me demonstrate it. First, the aim was to determine the onset of ozone-induced eosinophilic rhinitis in this case. And rhinitis and asthma go hand in hand in their disease. I'm going to start out in the nose and then end up in the lung. So we exposed these animals to 0.5 part per million ozone for 1, 2, 4, 9 days. We did nasal histopathology, morphometry, immunochemistry, and also RT-PCR. And we found that the lesion was in the anterior part of the nose, just like in the primate. It was on the lateral surface, very focal area in the nose. We then looked at nasal epithelial thickness, but also the inflammatory cells. I want to focus just on the inflammatory cells here, the neutrophils and the eosinophils. So after day 1, this is day 1, after a couple hours, and then after 24, and then we're looking at after, can't read that here, but this is one day at 24, and then at the 9 days. And the key is that the neutrophils are present. It's a neutrophilic type 1 inflammatory response early, but then it switches with continuous exposure to a type 2 eosinophilic response. And this is illustrated here that we have increase in epithelial thickness, but we have a switch in inflammation from neutrophils to eosinophils. Classically, a classic cell type of asthma. We then went on to look at gene expression, and that same thing. The bottom line is that we had a switch in the gene profile from a type 1 inflammatory response early after 1 day to after 9 days, it goes into a type 2 response. We then looked by using genetically modified animals. We knocked out the RAG2 gamma C animals, knocked out all of the lymphocytes, and actually eliminated the whole response. So we took out T cells, B cells, and these innate lymphoid cells, and all the responses in gene responses as well as in terms of inflammation, actually all went white, which was quite amazing for this really strong irritant fluid. But that just told us that lymphoid cells are important. And then we went on to look at innate lymphoid cells. In this case, what we used is RAG2 knockout animals. And these animals, we were able to use these animals because they have T and B cells. I'm sorry, they don't have T or B cells, but they have innate lymphoid cells. While the RAG2 gamma C animals have no innate lymphoid cells. And when the animals that have innate lymphoid cells, the wild type or the RAG2, all the lesions came back. And the gene responses as well. So in this study then we showed that in the nasal study that the innate lymphoid cells were associated with these lesions. If you took them out, the lesions went away. So then we carried it on in the lung, same approach, and where we eliminated the innate lymphoids, or all of the lymphoid cells, and some of the animals have just the innate lymphoid cells, and the same exposures of 1 day or 9 days. And just to illustrate here, is that with this animal model, we were also demonstrated, if we look at the different inflammatory cell types, the eosinophils, that in the animals with innate lymphoid cells, you actually have eosinophil response characteristic of asthma, also lymphocytes, but in those without innate lymphoid cells, that is all gone, even in the face of continued exposure. And so we also looked at it after 1 day and found that that wasn't the case. So just as we had predicted, is that innate lymphoid cells aren't important in the very beginning, but when they go to the asthma-like phenotype, they're the major drivers. So we also showed the mucus cells in the airways were associated with innate lymphoid cells. They were present in those with the innate lymphoid cells, but not with the ones that didn't have them. And we further went on then to look at different gene expression for mucus and for inflammation, the same thing was demonstrated. Then we used we also wanted to use flow cytometry to understand whether or not these animals had increased numbers of ILCs. And so the animals that have the RAG2 animals that have ILCs but not T or B cells, when you're exposed to ozone, there's an indication that it may increase, but we didn't really demonstrate it. We did demonstrate there were no ILCs in the lungs of the animals with genetically modified. But then we went on to actually treat the ILCs with an antibody that knocks out innate lymphoid cells. So we knocked out the innate lymphoid cells. They only make up less than 2% of all the cells in the lung, but we were able to knock them out. And the phenotypes, the increase in eosinophils that we saw, that was eliminated, as well as the mucus in the airway. So we think that these cells are very, very important in the response to air pollutants, and they are important, we believe, in this non-atopic asthma. And this was used. We demonstrate this in the mouse. But I think the key, after I showed you all the things where there's differences between the large animal and the rodent, that really needs to be carried back into the primate model. And this now is one of the areas of focus by Summers researchers now at UC Davis. The real thing is the treatment, though, the translation of that. And so one area of translation that might be very helpful for asthma is actually to look at how to deplete or to stop the effects of these innate lymphoid cells. And it has to do into a model that truly mimics the human condition. There are some pharmaceutical companies that are actively exploring that, these alarmins that have been demonstrated. So we were the first to demonstrate this in the mouse. There's 2 other labs that have verified that now. But I think the key is here again, these large animal models, before we can translate it into prevention or treatment. So I close here with some suggested readings. And those readings are really, I think, if you haven't read the book yet, thank you for being late. An optimistic guide for the age of thriving in the age of acceleration. I think you'll find this extremely great book. It touches on a lot of areas of where we have lost boundaries, and now we have to, if we're going to survive and adapt to this age of big data and how to translate it, we are going to have to work together on that, and we have to have time to really, really think about what we're doing. That we're not just generating data, that we're not So this is also a good book by Richard Parker on that. If you're interested in laboratory primate, we have a couple of chapters in this book. It's a good one probably to start. But I just want to close. This is an example. And certainly, I think that maybe an approach is to looking at farm animals or other large animals and disease. So thanks for your time.